Hi guys, good morning. In yesterday's class, that is of DNA replication, many of you had problem in understanding how exactly the end replication takes place, and that is mainly because of the marker problem. So today I have come up with one solution to this. I hope you will understand it. And in this only I will be dealing with how exactly the telomere replication takes place. So let us start with it. So the main problem in case of eukaryotic DNA is this eukaryotic DNA is having the linear nature, whereas the in prokaryotes it is in the circle form. So there the problem of end replication is not arising, but here the main problem is when I am removing the last primer. Say for example, this is your last primer, and once we reach the TRE region, TER region, the TUS protein will recognize that and it will stop the RNA polymerase from moving forward, and that's why the primer will be removed. So after removal of this primer, if you see the daughter strand which is in pink color, so much nucleotides, for example here there are 3 nucleotides in the parental strand, but the daughter strand is not having these 3 nucleotides. So what it means is after I am replicating each cycle, some amount of nucleotides are lost, and that loss leads to ultimately loss of the genetic information. So the message is not passed correctly from generation to generation. So to avoid this problem, the cell utilizes one enzyme called as telomerase, and what this telomerase does, it recognizes exactly the telomeric end. So in eukaryotes, the telomeric end is mainly meant for protecting the inner DNA sequences, and that telomeric end is made up of repetitive sequences which code for no particular gene. So here they are not coding for any particular gene, but they are protecting the other parts of the chromosome or the inner DNA sequences which are having the message for protein production. Right. Now what this telomerase enzyme do, it will recognize this telomeric DNA sequence which is starting from 5' to 3' in TTAGGG, which is repeated nearly 100 to 1000 times, and this is specifically in the human being. So each organism will have its unique sequence. Right. Now what this telomerase enzyme actually, so telomerase enzyme, actually most of the enzymes are proteins, but there are also certain RNases which is acting as a protein. And what is the main nature of this telomerase is it is made up of both protein and the RNA part. So it is having two components. One component is a protein component which is going to act as an enzyme. It is going to catalyze the polymerase reaction, and the second component is RNA, and this RNA will provide template, right? So we need in the normal DNA replication the parental strand itself act as a primer, but here the RNA component of the telomerase enzyme will provide the template. So understand this point. Protein part of telomerase is acting as a catalytic, and the RNA part is providing you the template. So now if so in this slide we can see the organisms and the telomeric ends present in their DNA sequences. In Arabidopsis it is TTAGGG. In human beings it is TTAGGG. Right. So and you can check the list. So this DNA sequences which are present at the telomeric end are species specific, in the sense each species will have its own telomeric region, and these sequences are conserved in nature. Fine. So now if you see, this is the telomerase enzyme. Here it is showing its two component. One is the RNA component which is providing the template, and another is your protein component which is behaving like a reverse transcriptase enzyme. So telomerase is also called as reverse transcriptase and the RNA dependent DNA polymerase. So actually it is a DNA polymerase enzyme, but it utilizes the RNA template, okay, and synthesizes the DNA. That's why it is called RNA dependent DNA polymerase. Whatever normal polymerase enzymes are there, they utilize the DNA only and they synthesize the DNA. That's why they are called as DNA dependent DNA polymerases. But your telomerase is a reverse transcriptase because it is using the RNA to synthesize the DNA. Okay, and the another is, it is also called as RNA dependent DNA polymerase. Now if you see this sequence and compare with the telomeric end of humans, in humans it is TTAGGG. If you see here AAUCC, it is RNA, so it will have U. So now this A will pair with this T, this A will pair with this T, U will pair with A, and this CC will pair with GGG. So what they will do, this region will form the complementary base pairing with the parental strand. We'll see that on the next slide. Now I have just shown you the last end part of the telomere, right? So here you can see this is your parental strand, this is your newly synthesized daughter strand. After removal of the primer, this much strand is overhanging in the sense it is not having its complementary strand in the daughter DNA. Now why it is not there, we already seen, because here there is no 3'OH group, so DNA polymerase cannot extend it, and hence the process of replication will halt here. Right, now what this telomerase enzyme will do is it will bind at this region, you can see here, and as I explained on the previous slide, it will form the complementary base pairing. Once it forms the complementary base pairing, now look carefully, this is your telomerase RNA component, and here it will provide as a template, and your parental strand is having the free 3'OH group, so this will act like the primer, right? And here the new nucleotides will be added. So new nucleotides are added at this region, and the telomerase is actually providing the template. Now it will extend in this telomerase is also having the polymerase activity, so it will extend this strand. Now you can see here it has added the new nucleotide. Our actual end was up to this G nucleotide was present in the parental DNA, and GTTAG, whatever is added, it is newly synthesized using the telomerase enzyme. Now how long it will go on adding the nucleotide is, and unless it reaches the 100 to 1000 base pair. Now once it has finished replicating this strand, what it will do is it will shift towards the 3' end, that is it will move from 5' end to 3' end, and again it will, so original position was this, now it has moved towards this direction, and you can see once again it will form the complementary base pairing, and the remaining part will act as a template, and here again new nucleotides are added. So fine, we have synthesized some, say for example 100 nucleotide long sequence on the parental strand. Now we don't need this telomere, so now this telomerase will leave the site, and after that what happens is, here they are given DNA synthesis by 2 rounds of telomerase activity, right? Now what we'll do is, here now the DNA polymerase enzyme will come, and that DNA polymerase enzyme, before that a primer will be synthesized here. So I'll show you, wait. So here the new primer will be added. Okay, so this is its 5' end, and this is its 3' end. So 3' end is providing you a OH group, and 5' end a phosphate group. So now if you see, this parental strand will act as a template, and here we are added new primer, so we can easily synthesize the new strand, and we can complete our original DNA information. So by this process what exactly we are doing is, we are getting the complete replication of the parental DNA in the daughter DNA. So once we have finished this, again this primer will be removed, because always a RNA primer is removed. We have removed this primer. After removal of the primer, again we will have some overhanging sequence, right? But now try to understand, this overhanging sequence is a extra sequence which was not present in our original parental DNA. So even if we don't replicate this sequence in the daughter DNA, much difference will not be produced, because already by using end replication we have completely replicated our DNA sequence which was present in the parent. Now whatever overhang is there, that is the extra sequence. Now once we have produced this overhanging sequence, what this overhanging sequence will do is, it will enter into the daughter strand. That is, it will move inside the daughter strand which is newly synthesized, and it will fold back on itself, forming a T loop like structure or the D loop like structure. Why? Because if we keep as it is linear, it will produce the sticky ends, and those sticky ends may cause the further problem by sticking together. So to avoid that problem, this overhanging sequence will form a loop like structure which is generally called as a D loop or the T loop, and in this way it will protect the inner sequences of the chromosome or the other DNA sequences. Here we are given another is, if you see in mammals, the telomerase activity is limited to immortal cells which are like stem cells or germline cells, and the main are tumor cells or the cancerous cells. So this telomerase activity is immortal, in the sense, the cells which are having this telomerase activity, there will be never a loss of information or shortening of the chromosome after each round of replication, right? But the absence of telomerase activity in the cell results in progressive shortening of the chromosome ends as the cell divides. So if telomerase activity is not there, after each round of replication, certain amount of information is lost, and thereby limiting the number of somatic division. So as we say, after certain age, the cells stop dividing or they lose their division activity or division capacity, and they become stagnant, and that is the reason for that is your telomerase. Because if telomerase is not there, we cannot protect the ends of the chromosome. So after certain stage of life or growth, it's the cell loses its division capacity. But if you see the tumor cells, they are immortal cells, and the one of the reason for that is your telomerase activity. So those tumor cells are having the very high activity of telomerase, and as a result they they never lose their information, and they go on replicating and go on dividing continuously. So nowadays the another main clinical approach is to delimit or to stop the telomerase activity in the tumor cells, so that their growth will be limited, like how many times it will divide, that might be limited. So that is another clinical application of the telomerase enzyme. So with this we have finished our end replication process. I hope you have understood it. So watch it and tell me whether you have understood it or no. Okay, thank you, keep learning and keep growing. Have a good day.